Please.
Okay, thank you very much. Good morning, everybody. Today I will present about my research. Okay, here is my research framework. So we interested in the engine of bovine coronavirus. So we use the genetic engineering method to evaluate them and express the protein. And then I bring the recombinant N protein to do the develop indirect ELISA. Okay, and this is the outline. Okay, and about the objective is to develop an ELISA for antibodies detection against bovine coronavirus. Okay, I will review about coronavirus. Coronavirus is a member of the order Nidovirales, family Coronaviridae, subfamily Coronavirinae. It's an enveloped virus with a positive single-sense stand, non-segmented RNA genome, and which are nucleocapsid of helical symmetry. Coronavirus virion has pleomorphic to spherical in shape, about 80 to 200 nanometers in diameter, and the genome size is about 26 to 32 kilobase pair. So the coronavirus is the largest RNA virus. Okay, coronavirus primarily infect the upper respiratory and gastrointestinal tract of mammals and birds, such as SARS, MERS in human, FIP in cat, PED in pigs, and IBV in poultry. Okay, and this picture shows the bloody diarrhea is a few cases in my country. The bloody is like a flaky bloody diarrhea. Okay, and about bovine coronavirus, it's rapidly distributed and incubation period is about 3 to 8 days. And the primary route of entry is via the fecal-oral route. And about host strain, all breeds and age of cattle are hosts. And the problem is about economic loss, is poor production and high decreasing milk production, and report in many countries and is widespread with high seroprevalence in adult dairy cattle. So about bovine coronavirus in Thailand, study of bovine coronavirus infection among dairy cattle are limited. Now we have just only 2 papers about bovine coronavirus in Thailand. First is from 1992 report prevalence of bovine coronavirus antibody in Baotang milk samples at 93% in Saraburi province in the central region of Thailand. And first report of bovine coronavirus molecular diagnosis of dairy cattle was detected in central region Thailand. And for genome organization and schematic drawing of bovine coronavirus, this picture review is genome include 13 open reading frames of coronavirus, and the engine is represented in the cyan color or ORF4. And this picture shows the 5 major structural proteins of bovine coronavirus. And this is the nucleoprotein and RNA genome. This is my gene that I interest. About nucleocapsid or N protein of bovine coronavirus, it's a 60 kilodalton phosphoprotein complex, length 1,347 base pair, and have 449 amino acids. An engine of bovine coronavirus has highly conserved sequence. The major antibody binding domain was also shown to be present in the N protein of SARS. And the N protein of IBV has many linear immunodominant epitopes. And in SARS, the anti-N protein antibodies were shown to be present longer and were more abundant than antibodies against other structural proteins. Okay, and next topic is about materials and methods. Okay, first about sample collection. Okay, we collect sample from the west and center region of Thailand, about 231 serum samples of dairy cows in 27 farms in western region and 6 farms in central region of Thailand. And then we collect the 40 fecal samples of dairy cows in 3 farms in western region and 3 farms in central region of Thailand. Okay, and then I bring the feces to the RNA extraction, and then I do the RNA synthesize to BcDNA. And then primer design and engine cloning for almost full range of the engine are predicted about 1,269 base pair of amplicon were amplified by forward primer and reverse primer. The purified PCR product was ligated into the TA cloning vector, okay, and then transformed into a competent E.coli strain JM109 using the heat shock technique. Okay, then the next method is subcloning of N gene into expression vector. The N insert plasmid and PQE vector were digested with BAM-H1 and HindD3 restriction enzyme, and then we ligate together and transform in the E.coli strain M15. And then we do the protein expression in E.coli containing of ampicillin and kanamycin, and divide into groups. Group 1 is the IPTG group. It's NPQE-HDL at IPTG 0.25 millimole. And the group 2 is a control group. It has only the PQE-HDL vector. And after protein expression, we do the protein purification by separate the supernatant and pellet, and then bring the pellet to lyse by lysozyme, and do the partial purification by urea, and do the affinity chromatography containing nickel NTA. And then we check the protein by Western blot analysis and biological function evaluation by transfer the recombinant N protein to nitrocellulose membrane using transport depth semi-dry, and do Western blot with anti-His antibody, and then do the immunoblot. And the last is an enzyme-linked immunosorbent assay establishment. And here is about the stable protocol that I try in my lab. Okay, and the next subtopic is result. First, about engine cloning. After we have we collect the feces that have the bloody diarrhea, then we do the RNA extraction and cDNA synthesis and use PCR using PNF and PNR primer, and we get the diarrhea cattle fecal sample in lane 2. This is show the PCR band. And then we send to the sequencing and use the RNA gene in the TA vector and RNA gene in PQE-XT vector. Okay, and this picture shows the pellet fraction that have the extra band of protein that I expressed. Okay. And then after this Western blot with anti-His, then we check which immunoblot with the positive serum and negative serum. And we can see the protein from supernatant fraction react with positive serum in lane 2. Okay, and about statistical analysis, the cutoff value of the recombinant N ELISA from the mean OD 450 and to standard deviation of the negative serum was 0.049. And the percentage of sensitivity and specificity was 84.8% and 96.3% respectively. And about coherence kappa value was 0.71. And in this table, by using 3 positive dairy cattle sera and 3 negative dairy cattle sera in quadruplicate, the intra-assay CV was indicate the range of 2.33% to 13.33%, and the inter-assay CV was in the range 4.48% and 18.75%. Okay, and about discussion, percentage of sensitivity and specificity were sufficient to detect bovine coronavirus infection, and the low to medium of percent CV for the intra and inter-assay value warrant the repeatability and reproducibility of the ELISA. And about the coherence kappa value, reveal substantial agreement between the recombinant N ELISA and the commercial ELISA. And have some paper, they success to express the complete N gene of IBV in prokaryotic expression system and develop ELISA. Okay, and the last about conclusion, this research was success in express the partial N gene of bovine coronavirus in prokaryotic expression system and develop indirect recombinant N ELISA in Thailand. And I would like to thank the Kasetsart University 72-year anniversary graduate scholarship for the financial support, and the biotechnology and serology laboratory in KVDU for the facility provided. Okay, thank you everyone for attention.